Good afternoon, everyone. My name is Vikas Bansal, and I'm from the University of California, San Diego. And I'll be talking about haplotyping human. So here's a brief outline of what I'm going to talk about. So I was asked to give a tutorial, and so I'll describe how we can do haplotyping or haplotype phasing, or in sequencing world, it's called haplotype assembly from sequence data, and the algorithms available for doing haplotype assembly. And also I'll talk about the sequencing technologies that allow us to do haplotyping. And finally, I'll mention how or why we should do sequencing-based haplotyping as compared to statistical haplotype phasing using a population of individuals. So just some background, everyone's familiar. Humans are diploid. We have two genomes inside every somatic cell, and one genome inherited from the mother and one from the father. And so if you look at any specific region of the genome, there are some positions where the two genomes differ. And if we do sequencing or genotyping, we can get information about the genotypes at each of these sites. And genotypes are useful because you can test genotypes for association with phenotype. However, for many kinds of analysis, we need information about haplotypes. So what are haplotypes? Haplotypes are basically the sequence of alleles at these sites that differ between the two chromosomes on a single chromosome. And for any given genotype configuration, there are an exponential number of possible haplotypes. So why do we need haplotype information as opposed to just genotypes? Well, there are many cases where haplotypes are more informative than genotypes. One example would be, let's say you have a situation where one allele at a site produces expression of the gene. So it's maybe a mutation located in the upstream of the gene, and you have a mutation in the coding region of the gene that causes a change to the amino acid sequence. So depending upon whether these two changes or mutations are present on the same haplotype or different haplotypes, you could have very different phenotypic expression of this gene. Another example relates to trying to identify disease-causing mutations in recessive disorders. So sequencing is very commonly now used to identify disease-causing mutations. And if the underlying genetic kind of model is recessive and the individual's parents are unrelated, then the most likely explanation is that there is a gene in the genome that has two rare deleterious mutations. And so when we do sequencing, we'll find the gene, we'll find that this gene has two mutations, rare mutations, both are deleterious. But unless we know whether these mutations are present on the same haplotype or on different haplotypes, we cannot say whether these two mutations are causal for this recessive disorder. And these kind of compound heterozygotes, they are kind of common in the genome. So when you sequence an individual genome, you observe about 20 to 85 genes that have at least two rare deleterious coding mutations. And here's a plot from a recent paper where they looked at exome data from the 1000 Genomes project in two populations, one European and one of African ancestry. And they found that on average, European individuals have about 30 genes where you have two or more rare deleterious mutations, while your African individuals have 60 genes. So the point they're trying to emphasize is that when you're trying to sequence genomes, trying to find deleterious mutations under a recessive model, you need haplotype information to determine whether the presence of two rare mutations is likely to cause disease. So how can we get haplotype information? Well, there are classical approaches, two classical approaches highlighted here on the left. If you have information about the genotypes So for the individual that we want to get the haplotypes for, if we have information about the individual's parents' genotypes as well, then we can use simple rules of Mendelian inheritance to infer the haplotypes. For example, if you look at this site here, the individual is heterozygous with C and T alleles, and we see that the mother is heterozygous, but the father is homozygous. So which means that the C allele must have been transmitted from the father, and therefore by inference the T allele must have come from the mother. So basically we can infer the parent of origin for every allele and therefore get the two haplotypes. One limitation is obviously that parental information may not always be available, and second is that if all three individuals are heterozygous, then we cannot figure out the haplotype information at those sites. Another commonly used approach referred to as statistical phasing relies on the fact that in human populations there are the alleles at neighboring sites are correlated, and so if you look in short regions of the genome, you have limited haplotype diversity. So for example, if you look at haplotype information from a population of individuals at these 5 sites, we see that there are 2 haplotypes that are the highlighted in blue that are the most common haplotypes, and they actually explain the genotypes of the individual perfectly. So therefore, based on maximum likelihood, we can infer that the haplotype for this individual are these 2 haplotypes. Again, this approach is also not perfect. If you have a site that's not polymorphic in the population data set, for example this site here, then we don't have haplotype information for that site. And also, if you have high haplotype diversity in some regions of the genome, you will have multiple possible haplotype phasings, so you cannot infer the right answer with certainty. And this is a very popular approach because again, this doesn't require any experimental work. It's a purely computational method, and over the last 10 years or so, or actually 15 years or so, there are a number of methods that have been developed in statistical genetics community for this statistical phasing. And yesterday's talk about imputation, the idea of imputation also kind of relies on the same models used for phasing. Okay. So I'm going to, my talk is focused on haplotyping from sequence data, and so how can we do haplotyping from sequence data? Well, the idea is quite simple. You do standard DNA sequencing. You take DNA fragments from an individual's genome. You subject them to sequencing using any a standard high throughput sequencing technology, and you take your reads, align them to a reference genome, identify sites that differ between your genome that you've sequenced and the reference genome. And if you have reads that are long enough to cover multiple of these heterozygous sites, then each such read provides haplotype information for those sites. And here, we have labeled each site as the 2 alleles at each site as 0 and 1, just to make the problem easier to understand. So basically, this read that covers 4 sites tells you that the 0 allele at this site and the 1 allele at this site, and similarly the 1 alleles at these sites are present on the same haplotype. So if you have multiple of these reads, you can use computational procedure to infer long haplotypes based on the overlaps between these reads. So that's basically the principle behind haplotype assembly from sequence reads. And obviously the key ingredient for doing this is that the read should be long enough that they cover at least 2 heterozygous sites. So if you have reads that cover multiple heterozygous sites, you can have assemble long haplotypes. So to measure, measure the kind of the contiguity or the length of these haplotypes, we can use a measure that's used in genome assembly, and that's known as N50 length. So when we do haplotype assembly, ideally we want to have the entire chromosome as a single haplotype block, but we'll get these smaller haplotype blocks because at some point the reads are not long enough to link together these variants. So we can represent, what's the contiguity of the haplotypes using this concept of N50 length, which basically means that it's the length at which 50% or more of the phased genomic sequence is in blocks of that length or greater. And this is a measure that's also used for assessing genome assemblies. So now I'm going to talk about a little bit about the actual computational problem of haplotype assembly. So you can think about haplotype assembly as a problem where you have the input data is basically a set of reads, and each row in this matrix is a read. And again, we've labeled the two alleles at each site by 0 and 1 to make it easier to represent. Each column represents a variant, and each row has information about a subset of the variants. So for example, this read here has information about site 4, site 11, and 12. And our goal is to use the overlaps between these reads to reconstruct haplotypes that are the most consistent with the reads. And if the data is perfect, which means that we have no errors, then every read or every row in this matrix should be a perfect copy of one of the two haplotypes. So in that case, it's fairly easy to assemble the two haplotypes because there's only one. You can kind of build the haplotype starting from one read and keep on adding the read that's consistent with that existing haplotype. And so you can get your, the solution quite easily. However, real data has errors. And so in that case, the problem becomes more difficult. And people have defined this concept of a MEC score. It's the minimum error correction score. And it's basically a measure of how consistent the haplotypes are with the set of observed reads. And it's defined as the minimum number of base calls or entries in this matrix that need to be changed for each fragment or read to be perfectly matching one of the two haplotypes. Haplotype assembly, has a long history. It actually was studied first in around 2002, way before even sequencing, next generation sequencing technologies started to emerge. And it was shown that trying to find the best MEC haplotypes is a NP-hard problem, and it's also NP-hard even when you have some restrictions on the reads. And there are exact algorithms for this problem using simple dynamic programming, but their complexity is actually exponential either in the coverage or the number of rows covering a site, or it's exponential in the length of the reads. And real data has many types of error, not just sequencing error, but you can also have errors due to alignment. You can also have false heterozygous variants. And you can also have cases where a fragment or a read is essentially diploid, so it's a mixture of the two haplotypes. And so therefore, there are likelihood-based approaches for trying to model this problem. And the advantage of likelihood-based methods is that you can kind of incorporate all of these sources of error in the model, whereas the MEC optimization only considers single site sequencing errors. So about 10 years ago, we developed an algorithm for this haplotype assembly problem designed to optimize or find a solution that's trying to minimize the MEC score. And again, this is a heuristic because this problem is NP-hard. But the key this algorithm was very simple in principle. And there are two key ideas underlying this HapCUT algorithm. One is that it kind of models the inconsistency between a pair of haplotypes and the set of observed fragments using a graph. And the advantage of doing so is that you can translate or kind of use the concept of cuts from graph theory. So you have a graph cut, and we basically showed that negative weighted cuts in this graph correspond to updates to your haplotype solution that can reduce your MEC score. So which means that we can use well-known heuristics for finding max cuts in graphs to iteratively search for haplotype solutions that have low MEC score. And another advantage of this modeling was that we don't really use any information about the temporal ordering of the variants. So many of the previous algorithms kind of start to build the haplotypes, starting from position 1 and then adding fragments going from left to right. But these algorithms, they cannot update the haplotype. If you make a mistake, you cannot go backwards. In this approach, in graph-based approach, there's no, there's no ordering of the variants that's needed. So you can basically use graph cuts to optimize or to find a good solution with low MEC score. And this algorithm was developed, the motivation for developing HapCUT was this diploid genome sequence, the first diploid genome sequence that was done using Sanger sequencing and published in 2007. And so we were able to assemble haplotypes using this our method HapCUT on this data. And by comparing against several other methods, we showed that HapCUT haplotypes have lower MEC score than other heuristics. And actually this algorithm here, hash, which is basically a probabilistic or a likelihood-based version of HapCUT. So both hash and HapCUT had virtually identical MEC scores. Okay, so this was 10 years ago. If we kind of fast forward to today, 2018, we're living in a world where we can do whole genome sequencing with this Illumina technology for about $1,000. But question is, can we do haplotyping similar to what we did with these long reads? Can we do it with Illumina sequencing? And if you remember, I mentioned earlier, only those reads can be used for haplotype assembly or phasing that cover at least 2 heterozygous variants. And the rate of the density of heterozygous polymorphisms in the human genome is low. So we have about 1 heterozygous variant every 1200 to 1500 base pairs. So which means that very few Illumina reads are haplotype informative. So the haplotype blocks that we get from Illumina sequencing are fairly short. They have an N50 of about 1 kb. And so that's not really very useful for trying to use whole genome sequencing for getting long-range haplotype information. So, we can do simulations and we can see that obviously we need longer reads, but not just slightly longer reads, but reads that have lengths of about 10,000 base pairs. If we want to get haplotypes with N50s approaching 300 kilobases, and that's kind of like a number that will allow us to phase entire genes. So there are technologies now available, single molecule sequencing technologies. Pacific Biosciences is one of them that can generate reads that are of this length. However, these technologies, Pacific Biosciences sequencing is much more expensive than Illumina sequencing. So, and you cannot routinely sequence human genomes using this technology. So can we still do something with Illumina sequencing? So there has been a strong interest in trying to solve this problem of haplotyping from sequence data. And starting about 2010, several groups have shown that you can generate virtual long reads using Illumina reads. And there are many different versions of this idea, but the key principle underlying these methods is as follows. So you start from high molecular weight DNA, so DNA that has the length of the DNA fragments is about 50 or 100 kilobases or as long as possible. And you basically partition this high molecular weight DNA into pools, and such that each pool contains DNA that's equivalent to about 3 to 5% of a single genome. And why do we do that? Because it means that if a single pool has only that much DNA, then at any given location in the genome, you only have one of these high molecular weight DNA fragments with high probability. So you might have a small number of locations where you have 2 DNA fragments, but in most of the genome, in each pool, you only have DNA at each location from one single haploid long fragment. And if you have many of these pools, you can amplify the DNA in each pool using techniques such as multiple displacement amplification or MDA, or some of the first methods use phosphate cloning. And once you have the DNA amplified, you can do library preparation. You can fragment it into shorter fragments, do library preparation, and sequence each pool separately using like a short read Illumina sequencing machine. And once you take the reads from each pool and you map them back to the genome, if there was a fragment located at a specific location, what you expect to see is a high density of reads at that location, and this corresponds to the fragment that was present in that pool. And if you have data from multiple pools, you can use information about, so basically this set of reads here, they correspond to basically the reads from a single DNA fragment, long DNA fragment. And basically that's your virtual long read. And from that you can get long haplotype information, and using overlaps between the fragments from different pools, you can assemble haplotypes similarly as before and get fairly long haplotypes. So that's the idea behind this approach, where you can use Illumina sequencing but still do haplotype assembly. And there is this review paper published in 2015 where all these approaches are compared and reviewed. So those who want to look into that further, they can read this review. There's another approach, a very different approach for doing whole genome haplotyping, again using Illumina sequencing. And this was developed in 2013 at UCSD actually. And the idea behind this approach is that you use this proximity ligation sequencing method. So proximity ligation sequencing is a method developed to study the 3D structure of the genome. And the idea behind this is that you take live intact cells and you cross-link the DNA with formaldehyde. So DNA fragments or DNA segments in 3-dimensional space that are physically proximal to each other will get ligated. And you can do digestion and biotinylation and then ligation using a restriction enzyme. And what you get after doing the ligation is chimeric DNA fragments with a mixture of insert sizes. So each fragment here is basically a concatenation of 2 DNA fragments that were originally separate, distinct DNA parts of the genome. And the distance between these 2 fragments can vary from a few hundred base pairs up to hundreds of megabases. And here's a plot showing the distribution of the insert lengths or the separation between the 2 ends of these chimeric fragments. And we see that there is a peak at very short, about 200 or 300 insert length base pairs. But we see there is a long tail of very long DNA fragments going up to tens to hundreds of megabases. And using mouse data, we analyzed this data, and we found that 80 to 85% of these interactions that create these chimeric DNA fragments are cis in nature, which means that these DNA fragments that are being ligated together, they are coming from the same chromosome, not the opposite homologous chromosomes, which means that they are useful for haplotyping because they contain information about haplotypes on the same chromosome. So, but there are a small number of fragments that are indeed trans, and the frequency of these trans fragments or trans pairs increases as the insert size increases. And using that algorithm HapCUT that I described to you earlier, we were able to assemble this data into haplotypes that span entire chromosomes, and using standard Illumina whole genome sequencing. So that's kind of a novel approach, that leverages kind of a molecular library preparation method developed for studying the 3D structure of the genome for haplotype phasing. So as I said, there is a strong interest in enabling sequencing-based haplotyping, and over the last 7-8 years, different groups have come up with different approaches, and some of these I described to you already. And then we also have these single molecule sequencing technologies, including Oxford Nanopore, which now is starting to be used for doing whole genome sequencing. And all of these methods can enable haplotyping, and to kind of address the computational problem, because all of these techniques generate data that needs to be assembled into haplotypes. So a number of algorithms have been developed alongside these technologies. And in this, the interesting thing is that there are these different methods that generate data that's very different in nature, and the error profiles are also very different. So which is kind of why we have all these different methods that have been developed for haplotype phasing. For example, with the long single molecule sequencing reads, the main challenge is that the error rates of the reads is about 13 to 15%. So it's difficult to determine whether at any given position, what's the allele you have. For high C reads, the challenge is that the insert size of the read pairs can vary, from a few hundred base pairs to a few hundred megabases, and many of the algorithms that have been developed were not designed to kind of, be able to address this kind of very long insert read pairs. So in a recent paper, we've compared these methods on simulated data, and the simulations were done to kind of capture these different aspects of these different types of data. So coverage per SNV, so that's the read depth. How do the different methods scale with increasing coverage? How do the methods scale when the read lengths get longer, so you have more variants covered by every read? And finally, a simulation model designed to capture high C data, where we basically have the number of read pairs crossing or covering or spanning a single nucleotide variant. And what we find is that most of the methods don't work well except on a few types of data. For example, ProbHap and RefHap don't scale well when the coverage increases. Similarly, HapCUT itself doesn't scale well when the length of the reads is increased. But our recent method HapCUT2, that was also described in this paper, seems to work well for all types of data, including this kind of high C kind of data. So we've also compared these methods on real data sets for this one individual N8-12878. This is a well-sequenced genome, and it's one of the reference genomes that commonly used for assessing haplotype phasing methods. And for this genome, high-quality haplotypes have been assembled by the Platinum Genomes project. So you can compare your haplotypes assembled using a given method and a given technology to these high-quality haplotypes and estimate the error rates. And just as an aside, the accuracy of haplotypes is measured using this concept of a switch error rate and mismatch error rates. And that's basically if you have your ground truth or true haplotypes, and these are your estimate haplotypes, basically you count how many times the phase between adjacent variants switches between is incorrect between the estimated haplotypes and the true haplotypes. And from that you can calculate a switch error rate, and also a mismatch error rate, where mismatches correspond to single sites that are flipped between the true and the assembled haplotypes. So when we compare these different methods across these different data sets for this one individual, we find that the performance of these methods is somewhat similar across these data sets. But again, So, two of these methods don't work on this 10x genomics data. Again, because this data has very high coverage per variant, and 10x genomics data seems to provide the highest accuracy in terms of switch error rates. Yes.
Because is this phasing all heterozygotes, or are there some heterozygotes that are not being phased that are not included in these calculations?
Yeah, that's a very good question. So that's also another metric that this comparison is using the subset of variants that were phased by all the methods to just make it easier as a baseline to compare. But there are differences. Some methods are more aggressive in phasing, so they will even phase variants that have just one read or some. But if you compare across phosmid and PacBio, there's also differences in the fraction of variants that are phased between these different data types. Okay, so in the beginning, I mentioned that we can do haplotype phasing with using the statistical phasing methods, and so the question is why. And now we have larger and larger reference panels of populations, so which can improve the accuracy of statistical phasing. So the question would be, why do sequencing-based phasing? And so there's a recent paper out in PLOS Genetics where they've compared how the accuracy of phasing variants depends on the allele frequency of the variant, the minor allele frequency. And what they've found is that when you compare these sequencing-based methods, the accuracy of the phasing or the switch error rate, that's the y-axis here, is fairly independent of the minor allele frequency. And that kind of makes sense because when you phase using sequencing, the allele frequency of the variant doesn't come into the picture at all. However, when you do phasing with this statistical phasing, the allele frequency matters because you need more the observations of the allele, the minor allele, better likely to be your phasing accuracy. And so therefore, at least with panels such as the 1000 Genomes Project panel, all phasing methods seem to have much worse accuracy for rare variants. Again, this can be improved by having larger and larger reference panels, but again, the accuracy for phasing with sequence data also does not depend upon the ancestry of the individual. So for example, African genomes have higher rates of polymorphism, lower levels of linkage disequilibrium, and therefore statistical phasing methods have lower accuracy currently. But again, for sequencing-based methods, the accuracy is almost the same. So those are kind of two advantages of phasing with the sequencing data compared to statistical phasing. So before I end, I just wanted to talk about kind of something that I mentioned before. So I talked about haplotype phasing using this Hi-C or proximity ligation sequencing. One thing I didn't mention is that in this initial approach, only 20% of the variants could be phased. And the reason for that is the Hi-C protocol uses a restriction enzyme, and the sequence that leads to non-uniformity in the sequencing, the sequence reads. And so you have regions of the genome that are far away from the cut site for this restriction enzyme don't have enough coverage to be phased. Recently, the startup company called Arima Genomics has developed a new Hi-C protocol that uses a cocktail of multiple restriction enzymes, and it seems to improve the coverage uniformity. And we've also worked to develop a method that combines sequence reads and information from population reference panels to reduce the error rate of Hi-C haplotypes. Now I won't go into the details of this, but we've sequenced 4 genomes from the 1000 Genomes Project, as well as this Genome in a Bottle Consortium project using this new Hi-C chemistry. And what we find is that with this, even with without using the population data, we can phase about 90% of the variants instead of 20%. So that's almost a 4-fold increase. And by including the data from the population panels, we can phase 98% or of the variants in each genome. And also in terms of the switch error rates without using population data are about 0.5% to 1%, and they go down to about 0.1% when we combine sequence data with information from population panels. And also, these phasing results are based on variants that were identified directly from this Hi-C data itself, not using any independent source of variants. So basically, this protocol allows you to do standard, variant calling, phasing, without using any external, information. So to summarize, hopefully I've convinced you that haplotype phasing from sequence data is the most direct and accurate method for, phasing human genomes. Still, there is some work to be done in terms of cost and technology development, but there are protocols being developed and these long-read sequencing technologies that are kind of continuing to improve, so that hopefully in about 5 years' time, this sequencing-based phasing will become, the kind of the routine standard. And also trying to emphasize that computational algorithms are key. Even you have technology advancements, you need kind of computational methods that can leverage these advancements to enable highly accurate haplotype phasing. And so I just wanted to end by acknowledging people who have worked on haplotype phasing. So Peter Edge, who's a student in my lab, and my longtime collaborator, Dr. Vineet Bafna, and also Bing Ren and Siddharth Selvaraj, who kind of developed the Hi-C based phasing method. Thank you.
Questions? I had a question about number of haplotypes. So when you compare, this, you're comparing like your results with this like high quality data set. Did you have like the same amount of haplotypes restored, or you compare just a subset of them? Meaning that if you have sometimes, do you restore the same amount of haplotypes, the same number that you need, or maybe sometimes you can have like more or less, maybe you restore some haplotype that's not there?
So you're talking about this one?
Yeah. Well, these high quality haplotypes. So you know the number of haplotypes you need to restore, right? When you start.
Well, for one individual, you just have 2 haplotype sequences.
Okay.
And the number of sites is based on, how many variants were identified in the sequencing of this individual.
Okay.
And we're comparing our haplotypes at a subset of sites that were present in that gold standard data set and each of these data sets.
Okay.
Yeah, so sequence-based phasing can work, for model organisms, other organisms. In fact, a lot of the users of our method HapCUT are actually, applying it to non-human plants, wheat genomes, stuff like that. Even like more than diploid, there's polyploid genomes and so on. So yeah, definitely sequencing-based phasing can work for, any genome as long, and some of those genomes actually have higher rates of heterozygosity, so you can do phasing even with Illumina reads. Yes?
How do you feel from the process? So you get these haplotype blocks that don't overlap the entire genome, but presumably you're going to return a complete haplotype, and then how do you do the last step?
So the way haplotype phasing assembly methods work, they will return a list of blocks, and then the variants phased and for each block. So they don't, at least our method does not try to stitch together the blocks into a single chromosomal level block. But you can try to do that by using population-based data, but we've seen that some of the places where these blocks, where there's no, the reason for why you have blocks is that there are long stretches of the genome where there are no heterozygous variants. And so it's very hard to span these boundaries even with population data, because again, you don't have, you may not have, we don't see that there is strong linkage information spanning that desert of heterozygous variants. Yes.
So what's the current state of the art for N50 for some of these methods, with some of these haplotype assembly methods? And has anybody constructed something analogous to N50 for the gaps between the base segments? I mean, so we know what the distribution of sizes is there. You were saying that for many of them there's too far to get into the information.
Yeah, so the N50, like for PacBio, would be around 250 kb. Again, it depends on the read length, and for read lengths for PacBio continue to change. For 10x genomics, it's actually about 10 megabases. Again, because 10x fragments are, they can be more than 100 kilobases long. So that's the N50. And in terms of the gaps between the blocks, I mean, I don't know, like what's the total amount of, in terms of base pairs that would correspond to those gaps, because the N50 doesn't really consider, that in terms of base pairs, how much of the genome was not covered.